Welcome to Web Microbiology, podcast for Bio 204 at Allegheny College of Maryland. I'm Professor Kelly. Let's get started. Episode 7, Unknown Bacteria ID Lab. Welcome back. In this episode, I'm going to take a step back from Chapter 18 and instead talk about the Unknown Bacterial ID Dry Lab. Many students struggle with this assignment, and I hope it will help for me to connect the dots on what you've learned so far before you try and tackle it. Now, first, let me say that if you were taking the microbiology lab in person, you'd receive a mixed unknown sample from me and have to come up with your own plan to identify it. That's the expectation of this lab, so really you kind of have it easy. I'm giving you the test results without having to perform them, but it's up to you to interpret those results correctly and make the identification. I'll start by stating the obvious that really isn't always so obvious. You have to read the directions. Use your notes, reference things you don't remember or can't understand, and reach out for help from me if you have questions. Watch the videos referenced and print out anything you might need. This assignment helps you practice reading biochemical tests and learn the flow used in real clinical labs. It'll also help you study for Lab Quiz 3, and rushing through it kind of defeats that purpose. Now that all out of the way, the Unknown ID Dry Lab has a couple different parts. The first is designed to help you get oriented to a dichotomous key. This is just a fancy name for a workflow that uses yes or no answers to narrow down possibilities. You might not understand what every term or word means when you look at the dichotomous keys I provided, but you should be able to follow the logic, follow the arrows and the charts. If that's difficult, please take some time to learn how to read a flowchart. And again, you've got it easy here because some dichotomous keys are just big spreadsheets with tests on one side, organisms on the other, and you have to pick the tests in the right order. For this lab, the focus is really on interpreting them, so I've really given you a lot of those ordering processes already done. Now you want to follow the instructions in the lab first and have the Part 1 quiz open in the background. It's not really a quiz. Just the format works for entering the answers to the questions. You won't be able to answer the questions in that quiz without the information from the instructions. In Exercise 1, you'll learn some basics about dichotomous keys. Part 2 of this exercise introduces you to what I'm going to talk about in the next episode, which is the nuance and reality of clinical labs. So in other words, how do they know what to do with different patient samples they receive, and how does the process actually begin? The first steps are nearly always the same, though. Depending on the sample, several types of selective and differential media are used under different atmospheric conditions, that's aerobic, anaerobic, or maybe carbon dioxide environment, to isolate pathogens from normal patient flora, determine oxygen tolerance, and collect all that initial information. Now, sometimes these initial incubations and the information they give inform clinical decisions right at the beginning, especially for critical patients. So first steps might look something like this. A wound stab arrives in the lab. The lab tech streets it onto 2 blood agar plates, a McConkey agar plate and a chocolate agar plate. So we've got 4 different plates here. The McConkey is going to identify our gram-negative lactose fermenters. The chocolate agar allows really fastidious organisms to grow, and the blood agar reveals hemolysis patterns. Of course, we label all these plates, place some into aerobic conditions, some into anaerobic conditions, some into carbon dioxide incubators. They're all at human body temperature, and we leave them in there for about 18 to 24 hours. You should notice that after incubation, the lab should have been able to isolate pathogens and has already gained a lot of information. Then we do a gram stain, and microscopic observations are performed. Now, these first steps answer the most basic questions of a dichotomous key: gram status, cell shape and arrangement, and oxygen tolerance. From there, you select the specific key for that group of organisms. Now, in reality, a dichotomous key can be one giant chart with every single question on it. but it makes it easier to read and navigate by separating the workflows based on all this initial information about the unknown bacteria. So that's why you see a group of dichotomous keys and you choose the one that applies to the information you have so far. Now you'll finish up this exercise with some practice reading dichotomous keys. So download or print the keys I asked you to reference and take your time learning to read them. Now you'll find an image of an example dichotomous key using Guess Who characters that builds on the analogy introduced in the last episode. So look at this first if the real dichotomous keys appear overwhelming to you. Exercise 2 then gets into the nitty-gritty of the lab, beginning with your assigned bacterial unknown samples. Now each sample is a little different, but they all contain 2 pathogens to be identified. The lab does the initial inoculation, incubation, and isolation to provide you with a visual of the plated results. So imagine you're in the lab and the tech hands you these plates, and now you begin making your own observation. If you read the instructions carefully, you'll gain some clues to help you interpret the plated results, take notes to help with your identifications, and you have 2 unknowns, 1 and 2, to identify. Then you'll see images of the Gram stains. Imagine you're viewing these through a microscope and record the Gram type. Now, if you're really good at Guess Who, the key to winning is to pick questions that eliminate the largest number of possibilities on each of your turns. Microbiologists and lab techs work basically the same way. So your next task is to interpret the catalase and oxidase testing. At this point, you should have enough information to select the correct dichotomous key to use for each of your assigned unknown. Once you select your keys, you'll choose which test to perform next. So look down your key for each unknown and select the test that will help you reach the bottom. At this point, you won't need the answers to each test, so just choose every potential test you would need, and that brings you to the end of part 1. After you submit the answers, I'll review your results and provide feedback before you begin part 2. Exercise 4 of this lab is the interpretation of biochemical test results. And exercise 5 is the actual identification of the unknowns. These correspond to the questions in the part 2 quiz. Now, I'll provide you with visual results of several biochemical tests, and it's up to you to interpret them. You've done nearly all of these tests in either hands-on or a virtual lab, and I've tried to provide control results to help interpret tests that you may not have seen. If you do find yourself lost, look back on the Brightspace page for those labs because there's a lot of information provided there too for you about each test and their results. You will have all the results you need to identify both of your unknown bacterial samples. Fill in the names for each unknown and do not abbreviate. I'll check your answers and I'll provide feedback. An important note I want you to remember is that you have to identify at least one unknown correctly for this entire lab to count for a grade. You don't have to get it on the first try, but you also don't get additional time to make more attempts without a late penalty. So submitting this lab early can help you get the best grade possible. Please reach out to me with any questions. In the next episode, we're returning to Chapter 18 and the realities of the clinical lab.